All
We live?
right, we are live.
Um, Good morning, everyone. Good morning, good afternoon, wherever you are. Welcome to the stream of pump science worm experiments on a drug combination that we've been excited to test for quite some time now. I'm joined by Krister and Girish, both big names in the longevity space. And yeah, so what we have for you today is kind of like the experimental readout of several C. elegans experiments that we've run, which were run initially. We've now ingested the data and we're ready to display it all on the Pump Science website. And yeah, I mean, I guess we can get into what the drug combination does, but largely speaking, it's in the vein of chemical reprogramming. Krister, do you want to talk about what chemical reprogramming is?
Yeah, super great to be here, and also nice to see that you're back from your wonderful honeymoon, Benji. Lots of good energy, and now it's time to pump the science. And my name is Chris Kalpi. I'm the founder of the Rapamycin Longevity Lab, and one of the compounds that we have launched on Pump Science is the TREX token. And the TREX token is connected to a groundbreaking longevity intervention, which is called cellular reprogramming. And what is special with that longevity intervention? It is an intervention that was discovered around 2012, and there was a person called Shinya Yamanaka who got the Nobel Prize for discovering cellular reprogramming. And if we, for example, look at what he discovered, when we look at each cells, then there exists a little bit like a hidden rewind button within each cell. And the thing that Yamanaka discovered was how to press this rewind button in the cell. And when you press that rewind button, then you can make, for example, an old cell younger, get it to a younger state. So, and you can press this rewind button and de-age the cell so that it gets to age as 0. So it's like an embryonic cell state, which is super fascinating. So you can take, for example, an old skin cell, press this rewind button, and then it becomes 0, age 0. But the thing with this is that there are some problems with the de-aging cells to age 0, because when a cell becomes embryonic pluripotent stem cell, then it loses its identity. So if it started as a skin cell, old skin cell, and you press this rewind button too long, so it becomes an embryonic cell, then that cell isn't a skin cell anymore. And when you stop pressing it, then it can develop to all types of different cells. It can develop to a heart cell, liver cell, or even a skin cell back. But you lose the control of what type of cell it becomes. And that is a quite tricky thing. You don't want to press that rewind button too far. And that is also something which our intervention on Pump Science is looking at, that we partially press this button so that the skin cell does not lose its identity and become some other type of cell. It's still a skin cell, And so if it's a 60-year-old skin cell and you press this rewind button for a short while, then let's say that it becomes a 40-year-old skin cell instead. And so that is a super interesting research that we are looking at. And I will say that this is probably the closest thing that the research has come when it comes to radical life extension on a cellular level.
Mm-hmm. I think it's such an interesting thought experiment because, and what's really convinced me personally that there's, significant promise in this area of research is like two reasons. Number one is the jellyfish that are basically immortal. I forget the name of the species, but they can basically, at a certain point in their age, just immediately become young again, right?
Mm-hmm.
And so they're essentially immortal. I forget like how long they can live, but one day they can just decide that they're going to change their epigenetics back to a youthful state. So that I don't know, Girish, maybe the name.
Yeah. Well, no, the name, no, unfortunately I don't. But I know another really great model case is in the human, right? Like the female ovaries, they age alongside the female, right? So a 35-year-old woman, for all intents and purposes, has a 35-year-old aged reproductive and ovaries that have accumulated, these hallmarks of aging that we've defined, like DNA damage and mitochondrial dysfunction and gene expression changes. And what happens during fertilization is biology, evolution has this inbuilt process in which, that secret rewind or reset button that Krister was talking about before is pressed so that when the baby is born, it's not, 35-year-old cells. Rather, it's reversed back to a young, vibrant, extremely healthy state with literally damages reversed. So it's a fascinating thing. And this process happens several times actually during embryogenesis at different stages, not just fertilization, but a few weeks in. So, that's a proof of concept right there in humans. So if biology has built this system, this reset button in ovaries, our cells all contain the same DNA. What's different is just which genes are expressed. So we're now trying to figure out what are the right set of factors, what's the right recipe, right? Kind of like baking that we can use to do this in different tissues and, different biotech companies. Life Biosciences is concentrated on the eyes and macular degeneration. And then there's others that are looking at heart disease or liver disease. But, how can we really do this either on a systemic level? Wouldn't that be amazing? And there are some interesting pieces of data that you can do this in the hypothalamus and maybe get some broader reversal across multiple different tissues? Or do we need to kind of pick apart the body and do it organ by organ? That's going to be part of the fun exploration. And super excited about the applications and the logistical implications of using chemical partial reprogramming factors like what TREX is.
Yeah, and also I just want to make a little bit side note there is that when it comes to, for example, the big players in the field, there are, as Girish mentioned, quite big companies like Altos Lab, who is backed by Jeff Bezos, and we have Calico backed by Google. We have New Limit backed by Brian Armstrong, Coinbase, and we have also Life Bioscience backed by the world-leading researcher David Sinclair. And all these big players, they're looking at genetic cellular reprogramming. And that is quite natural to start looking at that area because Yamanaka, who got the Nobel Prize, he was also looking at genetic reprogramming. But the thing that we are doing on pump science now is to look at the chemical reprogramming. So we are using compounds instead of manipulating the genes to see if we can mimic the same effects. And if we succeed, then that is super groundbreaking research that we are doing.
I think
Totally.
it's interesting too. I'm not sure if you guys saw Elon Musk talk at the World Economic Forum where he was talking about longevity, and he made the remark of like all of this, all of our bodies seem to age on like a clock, and if you can figure out what that clock is, then you can reverse aging. And he's like, I've never seen somebody that has a young right arm and an old left arm. So I think all of this is very much like tied to even how he's thinking about combating aging. I know he mentioned he's not super deep in the space, but even just hearing his top line remarks, I was like, very, very first principles focused.
Exactly.
Yeah.
And just maybe last thing, kind of like what Girish was talking about with the aging ovaries, is even old men can make a young, healthy baby, right? So clearly it's physically possible to make a young version of yourself, right, to some degree. And so it's not that the ability is lost by any means.
No.
It's just that, basically evolution has decided that it wants us to turn over, right? But still the ability is there if we can harness it.
Yeah, exactly. And that is exactly the thing that the researchers have found, that they have looked at how does the biology work and how can we find intervention that could, for example, mimic those things so that we can, for example, reverse aging. And I know, for example, they have tested this in mice. They have taken an old cell in mice, reversed that cell to an embryonic cell, and then take that cell and put it to an egg or something like that. And then it gets to a full-grown mice. Take that old cell again from the mice, and then rewind it back to an embryonic state, and then repeat it step by step. And I think they have done it like 15 times or something like that to show that you can rewind the clock and the cells without creating any strange behaviors or side effects.
Totally. Shall we look at the data, ladies and gentlemen?
Yeah, maybe we can also just mention that how pump science work. If you can just explain where in the phase we are now, Benji.
Yeah, absolutely. So basically, each scientist gets to come up with an idea or set of ideas that they want to get tested, and those ideas are different, compounds, right? And they can be FDA-approved compounds, they can be research chemicals, they can be peptides. Really, it's whatever the scientist thinks can potentially be a life-extending therapeutic, essentially. And so what we do is we give them the tools, the funding and the infrastructure to be able to go run those experiments. And then we live stream the data onto our site as soon as it's been generated. And the tokens basically enable the research funding, the trading of them. So basically what has been tested here is this combination of chemical reprogramming agents that the team thinks can, basically extend lifespan. And so the first experiment that we run is on C. elegans, which are short-lived worms, followed by flies and then mice. And so, the stage that we're at currently with this compound is that it's just been tested on worms. The lifespan data has been gathered, and we now have it ready to stream. It was tested at 2 different doses. So we have like a low and a high dose that have been, it's actually not 2 different doses. It's 2 different conditions. But in one of the conditions, the coli were killed, and in the other one, they weren't. Maybe you can explain a little bit more of that, Krister.
Yeah, that is a very good thing to explain, because the diet or the food that you give to the worms, you can kill the bacteria in the food, or you can have live bacteria in it. And the thing is that in most common cases you have live bacteria in the food. But the thing is that when the worms get older, then the bacteria in the food has a tendency to start harming the worm. So that way it can reduce the lifespan of the worm. So it gets simplified, like it gets a bacterial infection in the gut, you can say. So if we, for example, give the worm killed bacteria food, then we can remove that parameter when we test the compound, so that see that if we, for example, use compound that kills bacteria, like antibiotic or something like that, then we can see, if we use live food, quite dramatic lifespan effects. I think you saw that, for example, when it came to what was the compound that you launched earlier this year?
Which one?
It was
Griffin Euro.
Yeah. Exactly.
You know, we launched them in 2024. It's already like 2 years ago.
Okay, yeah.
Just over here.
Time goes.
That's so crazy. It was that long ago.
It's 2026.
Yeah. And the thing there is that has an impact on the bacteria in the food, and that can increase the lifespan in the worms. And so the thing we wanted to do is to test 2 different conditions when it comes to cellular reprogramming in worms: live food and also kill food. And just one last thing here. The super interesting thing here is also that if we look at bacteria in the human gut, that because when we, for example, eat and things like that, we get bacteria into us, and we have a huge amount of bacteria in our gut microbiome. And there are some research that points out that when we get older, then that microbiome can start to create negative effects on the human body. You can, for example, if you get a leaky gut, then you can start to get strange bacteria inside your body, etc. And it can start to create lots of different strange inflammation symptoms and other things. So that research is also interesting to look at, but a little bit a side note there.
Awesome.
I was going to say all important context and background. I think even too for people, really what we're trying to do here is bring people into the research and into the science and show people the results. I know normally how things are revealed maybe be in like a paper or a preprint. And so it's really cool to create these moments around the data just to come together, share this context, have conversation, and then ultimately reveal the results. So it'll be cool to see how this all progresses as well. So hopefully we can keep people along for the ride.
Yeah.
Awesome. So the data's in. Should we check it out?
Yeah, it has been started to stream more.
Yes, sir.
Cool. This is the first time we see the data, so that it will be interesting to see.
Yes, sir. So what you're seeing here on the x-axis is each time the worms are imaged. So you can think of it as time, right? But basically over the course of the worm lives, we image to see whether the worms are alive or dead. And so what up here means is that the the worms that are receiving the drug are living longer than the control. And so what you'll see here is that for the experiment where we did not end up killing the E. coli, basically the food for the worms, the worms did seem to live longer, not by a ton. I mean, it's like from a mean lifespan extension perspective, it's just under 2%. The median lifespan didn't really have a difference. It was the same for both drug and control, but this is for one of the two experimental conditions. I will pull up the other one.
So this one was with lived food, or
let me double check that, but I think this is the one where where they're actually dead.
Okay, actually dead. Okay, yeah.
Let me see.
Because that is also interesting. We had, for example, a lab who has looked at the worm experiment who got more than 40% median lifespan. But it's that is the good thing, that we see if we can reproduce that result in another lab and see, for example, okay, what type of parameters did they use that we did not use, or did we use the same parameters, etc. And so this is let's but was it lived or killed?
These were the live ones.
These were the live ones. Yeah, cool.
And so
is that what you would
we
expect with the live food?
I think they did not test with live food. They only tested with killed food.
Okay. So
it will be interesting to see it killed.
Are you ready?
Here. Yeah, cool.
Are you ready? So much bigger effects with the killed food.
Much cool. And what does 5%? Okay, yeah.
But for median, it's 13%.
Mm-hmm. No, but that's interesting. And the thing here is that I think it's good that it was not a negative life extension. That is always something you're afraid of during a live stream, that you get a negative result. But even if you get negative result, that doesn't mean that it's the intervention does not work. It can be that the dose is wrong, or some type of other condition is not right.
And
so, yeah,
I have to nerdy worm-specific inside baseball hypotheses for why epigenetic or partial cellular reprogramming may be a little bit harder to achieve in worms versus mammals. So, I mean, the fact that we got a 13.1% increase in median lifespan is encouraging. Hopefully it guides directionality, and that's like kind of part of the beauty of being able to optimize dose and keep iterating on this at sort of crypto speed. So that's going to be, that's like one huge asset there. But one of the things that, or two of the things when I was working back in a C. elegans lab, I worked with the C. elegans intervention testing program and just went deep into the worm biology. And some of the things that kind of perplex me is, number one, C. elegans, like the way they methylate there's different marks you could put on your DNA to kind of fine tune how genes are expressed, right? And that's how these partial cellular reprogramming factors work. They, tighten up and knot up some areas of the genome, and they open up, like a bookmark, other areas of the genome so that, youth promoting genes, to simplify it, are more easily expressed. And those that are more like pro-genomic or pro-aging are more tightened up and balled up, so they can't express. So it kind of like reconfigures the genomic landscape, if you will. So C. elegans have like a really different process for actually like putting marks. So most of the epigenetic marks that occur in mammals happen on this cytosine molecule of the DNA, and C. elegans don't do that. They methylate a different area of the genome or nucleotide. So I think that's like one thing that's a little bit weird and a little bit different between translating like how partial epigenetic reprogramming factors work in mammals versus worms. There's like differences and nuances in terms of how they're actually working. So that could have something to do with it. But I think the stronger thing about C. elegans is that, there's pretty strong evidence that they don't exhibit very strong reprogramming as it is. And one sign of that is this whole, I don't know if you've heard transgenerational inheritance. It's this idea that, the stress of the grandfather can be transmitted down a lineage to the children and the grandchildren and the great grandchildren. And there's some, kind of signals within the literature that this happens in humans. There's super interesting studies and podcasts about, individuals that have survived extreme stress and famines and, Holocaust, like terrible situations in which, their genome is of their children are actually altered to have heightened cortisol expression or differences in metabolic fuel burning, and there's reason to believe that some component of that stress that their parents or grandparents experienced was actually passed down and preserved through the genome. With C. elegans, they're extremely susceptible to transgenerational inheritance. So like with mammals, with humans, you usually see this effect diluted out within a generation, maybe two. And with C. elegans, it just persists for, multiple generations, 5, 10, it can really persist really long. And part of that reason could be that they have less capabilities to, kind of turn back the clock and reverse some of those changes that were passed down to them. So that's another really interesting piece. Interesting story and piece of literature that people should definitely read into. I think there's a cool Radio Lab podcast on it as well for those that want to listen to something. But I think that could be something. It might just be a little bit more challenging in worms. I don't know. I think the reprogramming lab did see some bigger effects with their cocktail. I think, Krista, correct me if I'm wrong, something around 40%.
Yeah, 42 or something like that. So 48 maybe. Yeah.
So there's some optimization to do for sure, but those are some challenges in the way of worm biology, at least.
I was going to say, I had no idea they carried so much baggage, like historical baggage. It's very fascinating. How are you guys thinking about like the, like I know you said the optimization. So is this something from like a worm experiment that you would like to like try different things in worms again? Or are you thinking about, the more advanced animal models? As you think about flies, for example? Just curious to hear your guys' thoughts based on the results.
I think the thing that we would like to do is to look at flies because no one has yet looked at flies. So if we can get that data, that would be very nice. And also nice exposure for pump science that we are delivering unique data to the longevity field. So most likely flies. So it will be very, very interesting to see what that data looks like. But the next thing we are doing after this session is to analyze the data together with reprogramming lab, who Rapamycin Longevity Lab has partnered around when it comes to this intervention. The reprogramming lab is heavy researchers behind it. So and true believers of reprogramming and pushing this intervention forward in different ways. So exciting times.
Very exciting. Well, if you want to download the data, you'll just come here to the data section. Make sure you have the right experiment selected. It's 3 and 4. Let me check which one is 3 is the killed bacteria, 4 is the live bacteria. And you'll just come here to table and click download CSV. And this will allow you to download all the survival data. And we can send you the images as well if you like.
I was going to say, I dropped a message to Ben at Aura, who helped run these studies, and he's going to try and jump on here in a sec. So it could be cool to get his thoughts as well.
Yeah, if we give some background around that, Gillian or Ben, who is Ben Blue and what is Aura? And yeah.
Yeah, so Ben Blue is the CEO of Aura Biomedical, which is basically the lab that runs all of the C. elegans experiments that we're conducting. And yeah, they've basically built a bot that has kind of automated the process of running these lifespan experiments for C. elegans. So it's kind, you can kind of think of it as a grid where you know, kind of each cell is like a well where there's a different experimental condition in there. It's, you know, either the worms are receiving a drug or control, various doses, and there's basically a camera that sits on top of the well and just moves across the grid and takes pictures of everything. And they have, you know, kind of computer vision, machine learning models to figure out when the worm died, and, you know, without any sort of human labeling, can, you know, basically get survival curves. So we've been partnering with them. They obviously are extremely cost efficient because of all the automation that they have to execute all the worm experiments. And yeah, we're also working with labs that run fly and mice experiments as well. But yeah, the CEO of the you know, in our case, the worm lab.
Yeah, and Girish, who has worked with worms, is the worm expert here. I think you have worked with manual experiments, Girish, and it's quite time consuming compared to using a robotic system.
Extremely time consuming. We, you know, as part of the C. elegans intervention testing program, we had like 6, you know, technicians, grad students, individuals, you know, kind of picking away for hours and just like transferring hundreds, thousands of worms from one plate to another. And a big source of variability that we saw, because within the intervention testing program, what was cool is that the experiments were run across 3 different institutes, you know, Rutgers University, Oregon Institute, and the Buck Institute of Research and Aging. And, you know, we would be using the same drugs, same plates, same nematode growth media, the same light source on our microscopes. I mean, food source, everything was dialed down to the T, right, to make this as rigorous an experiment as possible. And time again, we kept coming back to the conclusion that one major source of variability was just manual error. Some people, when they picked up the worms, they kind of bashed them on the head when they were doing it, right? Some people were a little bit frustrated or doing it in the morning or late at night. Who knows how else they were, you know, intoxicated. Anything can happen, right? With these, with the different labs and human error and behavior coming into the picture. So being able to remove that and use these automated machines to do it is a game changer, because I mean, it just like takes out that huge variable that gets in the way of, you know, rigorous replication of these studies. And we've seen it. If you track the history of the publications from the intervention testing program, they've improved greatly, at least in terms of the error, replication error between labs. So, you know, huge advancements here. And I think amazing that Pump Science is able to do that, like across worms, across flies. Yeah, we're really off to the races with this product.
And I think just to point out the thing there, that Pump Science has built a super exciting pipeline here. So they used Aura for automatic worm experiments, and that way they can reduce the cost and fund research in a much efficient way. The same thing with flies. You have done in a great way. You have partnered with another company, and when it comes also to mice, you have also seen great ways of working there. So big thumbs up for what you guys are doing there in improving and trying to create a high efficient pipeline for longevity research. So yeah, thanks.
Yeah, you can think of it as almost like centralized research, decentralized ideas and funding. So kind of to your point, Krister, there's labs that we work with that run these experiment models for us, and the ideas and the funding come from a decentralized manner. So it's really cool to kind of see all of the ideas not only be able to be funded, but go through at like a high velocity because we have the infrastructure set up. So I'm super excited that you mentioned that this type of study hasn't been done in flies before. It'll be really cool to see the results from that and how quickly we can get that up and running and underway for you guys.
Yeah.
Ryan Bezecourt said, long live the science. Long live the science.
And Ryan is also one of the another developer team for the Raptor we can mention is on driving the research forward on pump science. So I can recommend to check them out as well. Super great team and doing great stuff there.
Yeah, he suspects. He thinks it'll work better in flies. So we'll see.
Yeah, let's see. We need to test.
Only one way to find out.
Yeah, no, but that's the real good thing, because many things when we find, for example, one intervention, we're guessing, okay, this may work, be a good intervention, but we really need to test. We need to generate data to see, okay, how does this work? And in some cases, it may work, not work in worms, but work in flies, or it works in all species, but not work in humans. So, yeah, we need to create a great machine for generating data on a high efficient way. So, yeah.
Amazing. Well, Margaret seems to be liking the data.
Love it. Yeah.
We got to get that fly experiments funded anyway. So
yeah,
let's make it happen, team.
Yes. Yeah. Let's do it.
Let's see. Ben, right on time.
Happy
Can
Friday,
you hear us okay?
Hello, hello.
Hey, Krister, Benji, Jillian, good to see you guys. How are you?
What's up? Doesn't look like the polar vortex is hitting Seattle.
We have a shocking bit of sunshine this morning. Here, let me close my blinds.
No,
I was going to say, this
you're
aura
glowing.
that I'm getting from you, Ben, is quite an aura.
Aura's aura farming, you know? That's our goal. Thanks for the invite to the stream, guys. It's good to see you all I think it's one of our first catch-ups in the new year. How's everyone doing?
So good. So good. And we just streamed the experiments from the chemical reprogramming agents.
Awesome.
Who can pronounce this?
Girish, I think, is good at pronouncing that one.
Guys, I'm glad I'm not the only one that stumbles over the word.
Transcyclopramine is I think it was. Correct me if I'm wrong here. I got to double check, but
it's
I think it was
FDA approved.
FDA approved intervention for an antidepressant, and shown to have some effects on reduced neuroinflammation as well. So there's some interesting properties there. But yeah, FDA approved, which is a very cool feature of the one of the ingredients in the chemical reprogramming cocktail.
Amazing. And then Repsox, which I think is 2 other drugs, right?
Mm-hmm.
Yeah. Awesome. So we streamed 2 different experimental conditions. One of them had the worms, the E. coli killed with UV, and the other one kept them alive. This was actually the case where they were killed. So it was, very prescient of the team to realize that would be, an important thing to do, because the data that we got from the 2 experiments is actually quite different. We got no lifespan effect in the case where the E. coli were alive, but we saw a 13% median lifespan extension in the UV killed bacteria condition.
Very cool.
Ben, any post-game thoughts there?
Yeah, I think it's really exciting. One of the things that is, I think, so cool to address at this early stage of drug discovery is the interaction of these complex emergent systems, right? So, reason we love this is it's the simplest multi-tissue animal, but it's also living and dying in an ecosystem just like humans are in the day-to-day experience. So, when we see stuff like this, there's a implication, and we always have to do additional research on it, that the E. coli may be actually metabolizing some of the interventions as they're getting to the animals, causing a potentially null effect under some conditions with live E. coli versus the treated animals living longer. This is kind of indicative of what sometimes people report with, let's say, the use of some different types of supplements where the gut microbiome actually may metabolize them before they're bioavailable to the human system. And so this is some of the complex biology that we start getting into in any sort of drug discovery. And that's why, the team's thoughts to do both in conjunction was honestly, a great step forward in assessing these molecules and interventions by the scientists that propose these experiments.
Yeah. No, but it's super interesting that just changing some conditions, you can get things right. And also, there are so many parameters that you can change. It's a little bit frustrating that we cannot change lots of different things and get get that data immediately. So, but the great thing with your technology, Ben, is that I don't know how many or dose experiments can you do in the warm bath for one batch?
For one batch, so we can do, generally in one setup, we launch about 20 interventions at a time, but we launch multiple of those each day, usually. And so, we're on the order of testing a couple hundred to 100 compounds a week. And so one of the things that is our internal strategy is to start very big across huge aspects of chemical space and just look for big signals, right? Something like some of the results from like the Omapal experiments, Krister, that you sponsored, where, that was part of just a screen. And now we honestly use that as a positive control because it increases lifespan so dramatically. And I think that's some of the strengths of the model. Is
that I just need to mention that the omeprazole compound, that was something that my lab, rapamycin longevity lab, discovered that it had lifespan properties in worms thanks to Bendu's warm bath technology. And as you mentioned, you use it all the time now, that compound.
Yeah, it's one of the strongest consistent modulators of lifespan in these animals that I've ever seen after 15 years of working with them. So
how did you I'd be curious, Christopher, how did you come to that idea? Like what is like the backstory there for what even led you to test that?
The thing was that I had a talk with the researcher Matt Kabeline, was it 2 years ago, a little bit more, and we started to talk about that it will be very interesting to do a big screening project around the different mTOR inhibitors, because one of the most promising mTOR inhibitor is rapamycin. It works great in different animal models, and but the thing that we lack in the field is to see, does there exist better mTOR inhibitors than rapamycin? So we set up a project together with Aura to screen 601 mTOR modulators. But before that project, I created like a small pilot project with 6 mTOR modulators or something like that. And omeprazole was one of those. And I got a big hit on that.
Yeah.
So yeah, so that was and the interesting thing was I went to a conference 4 months later after that discovery, and I was sitting in to eat dinner with conference participants, and then there were a researcher there who talked about, I have discovered one very interesting compound called omeprazole. But there is some lab, rapamycin longevity lab or something, who discovered it before us. And then there were some other guy. But that founder, rapamycin longevity lab founder, he's sitting beside you here. So and that way we connected and got friends. So it was quite fun story there.
I think what's cool is we spend a good bit of our time talking about the compounds and the science, but what shouldn't be lost on the community is the people behind the ideas. And all the folks on the line here, Krister, Girish, Ryan, Ben, are deep in the longevity space. And so these ideas that they're bringing to the table are really like top of the stack and ones that they are very eager to test. So just for folks who, might be tuning in, focused on the compound, I would also encourage you to pull up and look at the big picture here, because Krister, it's so cool to share that you were sitting at a table with someone who was like, dang it, this guy found it before me. Like, that's crazy. That is so crazy. And you have this idea here, you and Girish and partners that you're working with that are trying to get a compound to flies for the first time, like this study done in flies that hasn't been done. So what we're doing here, I know the pump science pipeline and infrastructure is set, but the ideas that are coming into it are not. So it's really cool to hear the story, and I appreciate you sharing a bit of the background, because that's why we're here, and it gets me even more pumped to do the science, because there's, these aren't just like, casual ideas. They're things that people really are eager to study, and you never know when you're going to hit the jackpot in terms of the longevity results on the next one.
Exactly. We need to create more of those damn moments.
Yes. I don't know. This in flies is going to be really cool to see. Like, I'm so pumped to get something to a fly study that hasn't been done before. And I know even too, like with Raptor, there's a lot of really cool work that the team is doing with, SMR28 and rapamycin. So yeah, there's just, I like I said, we come and we focus on like the set research pipeline that we have, but I would be like, really betting on the people. Like I would really encourage people to look at like your backgrounds, the people shepherding these ideas, because there's a lot of expertise on this call. I'm always humbled when we get on these calls to just be so close to these giga brains. It's amazing. It's really amazing what you guys are doing. I digress.
I think it highlights the power of decentralized science too, right? It's the beauty of bringing people with excellent ideas together in ways that actually get stuff done. So props to you, Jillian, Benji, you guys.
No, but as you say, Ben, we're doing something unique here. The thing, if we look at traditional research, let's say that we do some type of discovery, then it takes quite a long time to create the scientific paper, to get that published. It can take years before that data is out. But here we are talking like 1 month or something like that, and then you get that data out. Of course, that doesn't mean that we should stop writing scientific papers, but it's quite nice that we can, get the data out and get people working on things. So yeah,
I'm envisioning like a pump I'm envisioning a pump science conference where you guys are all sitting at tables, like coming up with your next, combinations that you're going to launch and yeah, like spitballing ideas that then you can launch right on the platform. Like that to me would be so cool to have the best and brightest minds and then to be able to launch them right away, like
Yeah.
I don't know. Benji.
Hey,
What?
that's a great idea. That
Yeah.
an actual conference is a great idea to get us all together.
It would be way
Yeah.
cool. And like launching ideas, just saying.
Yeah,
Just
like
saying.
having a round table, and then each person in that round table picks one intervention, which we launch on Pump Science.
Hackathon of sorts, like a proper hackathon, like a bio hackathon.
Jillian,
Yeah.
before OpenAI starts getting a slice of all the royalties that the IP developed, right?
Yeah. Yes, run as fast as we can. As fast as we can. Way cool.
Ryan, are you all right? Your voice sounds a little scraggly.
No, my voice is all messed up. I got whatever was going around, and I'm pretty much over the thing. And now I've got like my throat is like I can hardly speak. So it's like, and I've been on live streams, I've been in meetings, and my voice has not been resting. So yes, I'm also. Well, we'll see. North Carolina right now. We're about to get hit with that legendary,
Ice. Are you gonna get
ice
3 inches of
snowstorm
ice?
thing.
I'm just
Yeah,
wondering.
ice. It's like no longer snow. Now it's just deadly ice on the roads.
Oh.
But yeah, so we'll see. We'll see. But this is, by the way, I think this is incredible. Like, I was so excited for the data to come out. The fact that, as Krista was saying, we often either never see the data or it takes years before we see the data. And the fact that it takes weeks to see the data, a lot of the work, obviously, Ben, that you're doing is incredible. The fact that we can see it within weeks is like, it just blows my mind. In science, it's so often hidden behind walls. And so I know that, Christopher, probably wasn't what you were hoping for, the 13%. But to me, it kind of makes sense, like the stemminess of the cells of these C. elegans, that they don't really have real stem cells, right? And so I think in flies, I think that's where it's going to be way easier because flies are going to eat the food. And it like, does it penetrate the cuticle or not? Like there are all these other factors in worms. We found the same thing, by the way, with rapamycin and SMR28. Flies underperform, worms underperformed, and then boom, flies delivered. So
yeah. That's very interesting. Yeah, and that it works in different ways in different species. I don't know, Ben. Do you have any thoughts on that? You have been
In worms for a long time. Yeah. So I think it speaks to some of the different phenotypes that we see across different model systems, right? So even if we look outward, a step up from flies in complexity, mice don't necessarily always have translational results. People have cured many different diseases in mouse models. And I think it's what we use the system for, right? So we brought up omopolycib earlier, right? What these early translational models are good at is finding the big effects that we hypothesize are going to have a really, really strong impact as we carry up the line. And so what's cool about that is that occasionally we see those jackpots, right? And the bandit problem works out. It doesn't always. And so we see sometimes false negatives arise from the research where something doesn't necessarily translate. But I think one of the cool things about pipelines like this is that you can go and do these really, really quick. For us, what I like to do when I see something really strong, we've got a great team that takes it to human cells right away and looks at markers of senescence and markers of aging in human cells. Because I think that's one of the most strong proofs of principle of translational effect. And so similarly, if you can build this case that it works in worms, flies, and human cells, and then mice, you're completely off to the races in knowing you have a really strong biological effect that's going to get past the genetic diversity across these model systems, the different genetic pathways that are active, the different ecological niches that we have to think about, like when we talked about the bacteria earlier. And so while we may see some stuff not work in worms, and we may see some stuff work in other systems or not work in flies but work in worms, when we see the fraction of molecules that work across all of them, then we know that we've got something that's worth really pushing at towards getting to patients and people in a clinic, or to people interested in longevity medicine. And so what we're looking for is the thing that works across all of them.
Yeah. And one curious question, Ben. You said that you're testing the human cells. Is that expensive, or
It relatively no. I mean, it's more expensive than doing stuff in a worm. And the rationale for that, people can do high throughput cellular assays, but then you're looking at, a single cell or a single culture of cells, and you lose out on the context of, does this affect an animal aging in general, which is, I think, what we're all excited for when we think about finding these new discoveries. And so by having this pipeline that I think Pump Science and people that we're also developing, where you go and you use simple animals, worms, flies, then you have translational models like mice and human cells. That long chain of linkages for the positive hit is going to show us, what is the 1 in 10,000 or 1 in a million compound that's going to be, perfect for getting to people someday.
Yeah. I think, I don't know, Ryan, about you, but I think after the mice experiments, it would be probably interesting to look at the human cells, because, they're...
Seems like a pretty comprehensive snapshot.
Yeah.
And it's a very powerful package, right?
So that's a question I was curious about. Ryan, have you had a chance to accelerate some of these reprogramming discoveries into human cellular culture? I know some of these are based off existing human data too, right? Which is also giving you that endpoint from the very beginning. Where are you guys at in terms of that?
Yeah, Chris and Gerlish have been really exploring that. We have been very focused on the rapamycin SMIR28 combo. We're about to roll out another 4 programs. So we're looking at, I'll just kind of walk through what our big vision is. So our vision is, accelerate consumer longevity. And so it's basically 3 steps, or 3 main pillars. One is actually supplements. So what we're seeing is, and I'll share the specific supplements we're thinking about, but we're seeing that there's strong preclinical data and some clinical data even on supplements. So we want to bring that to market. It's basically the supplements I take. I take 10 different pills a day from 3 or 4 different suppliers. It's just a nightmare to deal with. So we want to get that stack done. We're testing that stack on Pump Science. And with you, Ben, we'll be, although I think actually with those, we're going straight into Drosophila and into flies, not worms. Then we're also testing, the off-label FDA-approved drugs, because we think we can go into humans much faster with that. So the strategy there is, okay, rapamycin is clearly one of them, but then there are a whole host of approved drugs that we think actually have some longevity, benefits. Interesting enough, people often talk about rapamycin and metformin, but the 2 of those might actually not be, they overlap too much. So we're going to explore that, but but a lot of people take rapamycin and metformin, right? And so that's the off-label drug. So that we would bring in a him and hers type model to the market. And obviously anyone else is welcome to do that too. There are other companies that are starting to do this too. Like AgelessRx, Anar is one of my favorite. And then the final is true innovation, right? Really bringing something truly unique into the marketplace. And so that would be, can you hear me okay, Ben?
Yeah, we can hear you.
You can hear me? Okay.
I'm so sorry. Ryan's audio isn't coming through for me at all. I didn't want to interrupt him because he looked like he was making a really good point. And I could hear people going, yeah. And I couldn't hear Ryan.
And I saw you going. Yeah.
Just say,
but you hear us now,
you said,
or?
I'm on record agreeing with you, because it looked like you're making a great point.
That was a head nod.
Yeah,
Good.
but you hear us now, or?
Yeah, I could hear you occasionally going like, yeah,
Weird.
during Ryan's
And you guys
thing.
hear me,
Everyone else,
but
I just can't hear Ryan for some reason.
weird.
I hear
Okay.
I hear you, Ryan. Good, but maybe
no.
it's something with your voice that
I'll log
is,
back in. I'll log back in. I'll see if I can.
No, but I think
Oh no. Well,
He'll be right back.
it might be on my end. Unfortunately, I have to run here in a second. But
Let's see. Can you hear me, Ben? Can you hear me?
Oh
I can hear
yeah,
you
now
now.
it's really good. Yeah.
So I won't repeat too much, but essentially 3 main pillars. Our strategy is consumer longevity, so supplements with preclinical and clinical data, off-label drugs like rapamycin, but much more than rapamycin, and then novel either combinations or truly novel drugs that are designed with AI. And so, one novel combination is rapamycin plus SMR28. Ethan worked on it 20 years ago at Harvard. And so obviously we co-founded Raptor together. And so that really is a molecule that he has been on his mind on and off over the last 20 years. And so, but that's not an approved drug. And so we would have to go through an approval pathway for that molecule, right? And so, but we think that there's an infinite white space, and especially now with AI. I don't know if you saw the recent competition. It was like some intelligent drug design competition, and like one of the winners used like Word. They're like, so out-competed all these like AI for science and whatever else. And so, I truly think that we are in a golden age for novel drug development. However, people are bringing bazookas when you only need like a BB gun sometimes, So, I think actually we are fit for purpose. All of us here on this call are fit for purpose because, like all you really need is a drug that actually works. It doesn't matter how you designed it, right?
Yeah.
If it came to you in a dream, I remember I talked to Kerry Mullis, the inventor of PCR. I don't know if you all remember Kerry Mullis. He was legendary. He won his Nobel Peace Prize. I don't know, 10 years ago, 15 years ago, I met him in San Diego. And, he basically tells this story where he was super high off of drugs, constantly doing acid and whatever else. And the structure of PCR came to him in a dream. Or either a dream or he was or he was tripping and it was kind of dreamlike. And that was it. That's how, and it worked, And so, unfortunately, Kerry Mullis has passed away, but like this is kind of, I think, the era that we're in. It's like, I'm super bullish on everyone here. I'm bullish on all of this stuff because no one's paying any attention to it. Like literally, I co-founded IndieBio. I know most of the VCs, the huge VCs in Silicon Valley. They used to come and hang out and talk to me all the time. I know I worked in drug development for a decade. I know many of the executives at Big Pharma. No one's paying attention to anything that we're doing. And when I talk to them, they're like, yeah, you're doing something weird with crypto and science or something. I don't know. And I'm like, look, just look.
Just wait.
Just look at what's happening here. This low cost science. This has literally been all of our missions for decades. And it's like here, it's just not evenly distributed yet.
Yeah.
Right. And
I think that's
all right.
what's.
We got Ethan. All right.
I was going to say these
Ethan's
AI
been on this
tools.
mission.
Yeah.
Yeah.
I'm so excited that you put it that way because like these AI tools, I'm very grateful to have them as a resource, but at the end of the day, you need real research. And so I think what's like you said, people are bringing bazookas and like they don't even have a BB gun. And like, you really need to give people the full suite of tools to actually take these ideas and test them. And so the low cost science, it's like, to even have something repeatable. Like I know Ethan has a ton of experience in rare disease where it was almost like rebuilding the infrastructure every single time from scratch. And it's like to just be able to have the infrastructure set and just plug in the ideas,
Exactly.
just plug in the ideas and just go.
Yeah.
It's crazy, but it hasn't been accessible or fast for a lot of people for a long time. But you, Ethan and Ryan and Krista, you guys would know far better. I mean, that's like a lived experience, very lived.
No, but it's
you
so.
can share your perspective. You went to. Sorry, go ahead,
Yeah.
Christopher. Sorry.
No, but it's so exciting times that we get, as I have pointed out, the high efficient pipelines which can start to generate the quick data to the field. That is so important that we get in place, because when that is in place, much will change, and that is the great thing with pump science, that you have created a great foundation here for Ethan, Ryan, and me, so that we can start pushing things forward. And I really love it. It makes my life much easier.
I'd be curious, Ryan, from your perspective, I think the biggest thing is like getting people to pay attention to like what we're doing. How do you think we could be better about showing people the value of like the low cost science and the speed to results? I think that's the part where it's like, we want to get even more people understanding what's here, and not like you said, people are like, what's this like crypto plus science thing? And I think it can almost cause you to lose the forest through the trees a bit. It's like, no, look at what it's actually doing.
I've
curious
learned
your
a
thoughts.
lesson. Yeah, I've learned a lesson, and then I'm going to hand over to Ethan because I think he has some really interesting insights. But I've learned a lesson. Sometimes you can be so ahead of like what people are thinking that they think you're an idiot, right? Like literally, it's just like, I remember when I came out with vegan dog food, people were like, literally, like, what the hell are you doing? We'll fund you to do a cancer company or a cell-based meat company. A vegan dog food company is ridiculous. So I said, fuck it. And I went straight to the people, right? I was like, I want to test my idea. I believe people will buy this vegan dog food. And they did. We had over 100,000 customers at Wild Earth. Wild Earth is still running. It got acquired by another larger company. And it still keeps growing. And we had vegan cat food too. I really believe we're almost at that moment. At some point, these big investors and these pharma companies are going to wake up and go, oh, wait, this thing has been going on all along right underneath us. We're worried about biotech leaving Cambridge, right, and leaving the US, going to China. It's been here all along. The answer is right here. It's decentralized science. They're not seeing it. And Ethan was just at JP Morgan, and he probably has a lot of perspectives on this, right?
I do. How's everybody
Oh boy,
doing?
I can't wait to hear this.
Is this the
He usually has the spicy takes.
Is this the after hours portion?
I mean, it's kind of like lunch break. I mean, it's a Friday afternoon, anything goes.
In Europe, it's spicy hour, I think.
Yeah, it's
5 o'clock
soon dinner time here. So quick comment there, Ethan, before I go.
Yeah, well, sorry for tuning in late. Ryan was texting saying, hey, you should jump in here. And so we know the feeling of an underwhelming worm result. So welcome to that club. But yeah, I guess dissecting the science here, that would be interesting in the days ahead. If you have, that's something that we've been doing with Raptor is doing just an extended meta-analysis. Our worm data is not so hot. And, we didn't have any performance data in worms. Maybe that's something you guys can try to do. You're taking pictures of the wells anyway. If you can get pictures of them, movies of them thrashing around, and you can get a movement score, I think that would be amazing, because then you could compare, performance to lifespan in a worm, in a fly, in a mouse, all in a kind of a normalized fashion. And I think that would be amazing to start to see what are the effects of compounds, combinations on the health span, lifespan axis as you go across evolution. And
I think you have that data, Ben, right, with movement and things like that, right?
Sorry, I'm having the issue again where I can't hear anything Ethan says, but I can hear everyone else. I don't know, it's me. I'm so sorry.
Okay, but one question, Ben.
Yeah.
He was asking you, Ben. Krista was asking you if you have the data for the worms for the reprogramming. If you have the movement data and things like that.
Yeah, it's all
locomotion data.
captured in the pipeline where we can look at movement over time, body morphology, a couple of other metrics of health and behavior. Yeah, so we can, I can talk to our team, and if folks are interested, we can disseminate some of that.
Yeah, we want to see the performance data because we have some strong predictions about how performance and lifespan are either coupled or uncoupled.
I'm so sorry. I unfortunately have to run. I've got a call at 9:15, and I also, hopefully I'm not interrupting Ethan, but it was lovely to see all of you. Congrats on the
Bye, Ben.
launch. And
Okay.
Nice meeting you. But Ben, we would love to chat. So we'll set up a time in the future.
Sounds great. I'll talk to you all soon.
Yeah, take care.
All right. Thanks for the invite, Jillian.
Yeah, of course, of course.
No, but I think, as you mentioned there, Ethan, that if we also can get the health span data combined with lifespan data, then we can start to draw quite interesting conclusions from that data and combine it with the different species. So you are doing really great work with showing the data in different ways and analyzing it together with the community. So big thumbs up for that, guys.
Yeah, Carlin was going to drop some new data on the effects of SMR28 rapamycin on older flies and how the effect actually is more prominent in older age. So we're really excited about, if we could have an anti-frailty medicine in fly, does that conserved in mice? Is that conserved in humans? So that's, something we're really excited. And that's not the data you see just on Pump Science. That's something where we have to scrape the raw data and then actually generate this additional analysis, which is fine. Like I don't expect Pump Science to be a full stack data visualization platform, but maybe actually there is a way over time to be able to have folks, play with the raw data. Someone who's a holder of one coin, or not even a holder, someone could just want to compare, across the whole portfolio of Pump Science to see, how do these different compounds behave relative to each other.
It's cool to hear how you guys are thinking too, because we obviously, even as you guys have seen with like mouse studies, there was like certain ways that we were thinking about it, or even Krista with the worms, where you're like, hey, can we change the food? So having these conversations with you all to figure out, as you're thinking about like a positioning or a product, or even like the picture that you're trying to paint, what are the additional data sets, or like how can we give you more information that you can cut a different way or tack on a different type of study at a different point. But to me, my gears are even turning on like, oh yeah, like could we add this in, or when do we put in maybe the human cell part that we talked about? Where does something like that make sense? But I think for us, we're here to serve and just want to follow the science that the market, that the drug devs want and that the markets want to see. So it all started with worms, and here we are. But that doesn't mean that this is it by any means. We can definitely have conversations to see where the pipeline goes from here. And we're actively thinking about the human component. I think at the end of this is like, how do you, at the end of the day, people want to know, what does this mean for me? Does something like this work for me? And so how can we, obviously I know that with, prescriptions, that's, it's a whole different animal. But as you think about the supplement space, how can we start looking at human studies and really getting additional human data? So a big part of, as we think about this pipeline, has always been design it end to end. And we're still, even as we get to mice, we're not at our goal is that that's not the end.
You should literally do races. You should have people race. Just like the flies do it. We don't have to negatively geotax. We can just, laterally geotax. But like, why the fuck not? I mean, honestly, it's like, if you want to make a comparison across species, you should do the same test as much as the organism allows you to physically engage it the same way. So like, imagine human racing, and and if you could do it where you can just organize your race, you don't have to go to a racetrack. You could just like, I don't know, or maybe you're kind of your phone kind of sets up, and you say, I want to run this distance, and so you look at your phone and it says, okay, you run from this point to that point. And then if everybody has that same app and they're like, okay, I'm running that same distance, and it can tell you the weather, blah blah blah, and then you basically can collect racing data like on your own, and then start to feed it to Pump Science. And then if you can connect the token price action and trading to the real-time results coming in, and then you could add other performance things, right? You could do the human, equivalent of grip strength. You could do pull-up challenge, push-up challenge, like all that can be decentralized. Data can be collected by phone and wearables. Like the moment for a completely decentralized, I mean, Krister, you were running you were kind of doing, no offense, but kind of like, the economy version of Brian Johnson's N of One, like Brian Johnson's first class. Brian Johnson has his own fucking jet, right? And so like you have the experience of recording data. Maybe you don't have a team of 50 doctors with probes up your ass and hollows around your dick to measure your erections, but like, you get what I'm saying? You ran an N of One, so you could productize your own experience with apps that exist today and enable fully decentralized human performance / lifespan, studies.
I was going to say that's like the vision of what we're hoping to build towards. Like, there's no reason why, as you think about like building that infrastructure with where we are today, with wearables, with how people meticulously track. I mean, there are people who track their supplements that they take every single morning. They have their wearable, they're tracking their, I turn on a workout thing every time I hop on the bike, like people are tracking bikes and reps and food and, their biometrics while they're sleeping. And so, Ethan, I think what you're painting a picture of is exactly the future that we're hoping to build beyond the animal studies, because that's the question. Like, people take product and stay on product when they know it's working for me. And I believe that there's a bit of a disconnect today where we have all of this data. I have more data about myself than I know what to do with, but what it's not tied to is like a product. So like every time I add something into my stack, I cannot answer what is this product doing for me? And so really bridging that, all this information with how you think about your stack, like what you're getting up and taking in the morning. Ryan was talking about like the fistful of supplements that you take. I keep adding another one into the mix, but I couldn't even tell you the incremental benefit that I'm getting. And it would be really cool to have that data for research purposes, but then also as a consumer, like, oh, this is what changed when I added this to my stack. I want to keep buying it because it's working. Like that is a fundamental question I think we as consumers in this day and age should have an answer to with every health and wellness product that we buy.
Yeah, and I think the interesting thing with this type of protocol, if we start to set it up, is that then we start to collaborate with each other in generating human data, and that will be super powerful. Because as you mentioned, Ethan, the experiment I did with rapamycin was like one N1 experiment. But the thing that my vision is like, I call it multi N1 experiments. And what I mean with that is that you have like one protocol that let's say that you take rapamycin on Saturdays on empty stomach, and then you do the running, let's say on Monday or something like that. So you have a quite standardized protocol, and then people can try this protocol, and then we gather in a structured way the same data connected to this protocol. And then we see, for example, okay, for Krister, it worked great. For you, Ethan, it didn't work as great. Why did Ethan not get as great result as Krister? Then we can, for example, start to also categorize people in different phenotypes. Let's say that you have a phenotype which works similarly to Ryan's phenotype. Then you can look, okay, what is Ryan doing? Ryan is using these things. Okay, maybe I can start using these things instead of using things that Krister is using, because he is totally different phenotypes than me. and then I think the real powerful things start to come. Yeah, sorry, I need to go now.
No,
my
we're-
We were hearing the
laundry.
dishes in the background, so we were
Yeah,
hearing that's a
yeah,
reminder.
You're done.
Gotta get off.
Yeah,
Clink,
my
clink.
my longevity intervention wife. Yeah, I need to take her now. So yeah,
Yes, yes.
yeah.
Well, everybody,
Take care,
thank you so
everyone.
much for joining. This was great. And yeah, I love the future that you guys are painting. And I think that's a future we're hoping to build very much at Pump Science. So I'm excited to follow up with each one of you and get a little bit deeper. And Krister, let's get those fly studies going. What do you
Yeah.
say?
Let's do it.
Awesome.
Yeah.
Let's do it. Awesome.
Take
care
Thank
now.
you guys. Ethan,
Bye-bye.
Ethan,
Yeah.
I want to hear about how the conference was too. I got to follow up with you.
Yeah. It's not all that amazing, but there's a few interesting. I think the major concept is, what I'm calling founder mode medicine. You're seeing all the stories of founders who are saying, my current startup is my health. And I think that's a trend that's going to start to explode in 26.
Yeah, it's really interesting. I read that. Is it What was it? GitLab?
Yeah, the GitLab founder Sid. Yeah,
GitLab
and
founder.
that
Yes.
story about him that Elliot wrote got massive readership. And then there's a guy who was working at XAI, Greg Yang, who said he has Lyme disease, and he said, I gotta stop working. It's killing me, and I'm going to try to fix my Lyme disease. So, the dominoes are falling. It's just like, oh yeah, think about it. There's a bunch of people, there's a bunch of millennials who have financial freedom, who are really smart, and who are entering the stage in their life when disease starts to creep in. And then it's like, boom, 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 perfect storm. It's like, hey, I'm a liquid exited founder. I've gone 0 to 1. I've mastered things I've never known before. And okay, this is the next thing is this, instead of product market fit, now I need to have therapeutic cancer fit. And boom, they just go at it. And like any founder would do the same thing with the same amount of capital. And there's just more of these millennial folks that are, it's just the ticking clock of aging, if you will. Yeah. So like, you're just going to see way more of these stories break out. And I'm personally working on one such case, and there'll be more to say about that at some point, but there's going to be a whole wave of like, essentially, if not billionaire cures, sort of like millionaire cures that are going to trickle down to the rest of the market, to people who are like them, who just didn't happen to be billionaires. So that one to end.
I mean, what you're describing, Ethan, I mean, you're describing where the next wave of biotechs is going to come from, right? I mean, you're basically saying there's probably going to be hundreds to thousands of new biotechs.
Well, no, and the beautiful thing is that each of them is going to be 100 times more efficient than the current companies. Because like Sid's case, it's like 7 different therapies or products, diagnostics, know-how, protocol know-how have emanated from his N of 1 journey, right? So just even though the drug product he made is not going to be like packaged up and like, hey, this is Sid's elixir, we're gonna sell that like GLP-1s. It's that every single one of these people going on this N of 1 journey to a cure, where it's 0 to 1, it's 0 to cure. They are going to just solve so many problems that the rest of the field is stuck on and will never fix in time or as cheaply. And now if you have like tens, hundreds, you don't need tens of thousands of these, but if you have tens, hundreds of these, centimillionaire to billionaires who are kind of going into their health to solve their problems, like that is going to and they're going to do it across they're all going to be affected by different, diseases. This one's going to be lung, this one's going to be skin, this one's going to be brain, this one's going to be immune. And then they're all going to just, each of their cure odysseys is going to just spawn so much, literally so much, a multiplier effect. So I think it's just going to rip people's face off. And it is diametrically opposite of big pharma. Right? Literally what you would say is, what is the opposite of big pharma? It's, what if I had an organization of 1, and instead of trying to cure the largest disease, it was just trying to cure that individual's disease. It's the perfect inversion of the pharma model. And yet it's the perfect bottom up alternative, I think, that we want, because if you have a bunch of decentralized N of ones, a bunch of decentralized Brian Johnsons who are actually sick, then all of a sudden you have this cure army. And with not that many of them, you're going to have massive multiplier effect.
I thought it was so fascinating reading the GitLab story, because it was like, even when he was like, I could get all of these like diagnostics. What I couldn't get was the information out of like the hospital system. And I thought it was crazy. Like, you think about this idea in blockchain of like self-custody and like self-custodying your assets and your, self-custodying your financial information. Like, the fact that there was this like information that he couldn't even self-custody to decide what to do with to get to the cures. It just like, I don't know, it was something that like, until he went through that journey and realized, how risk averse these hospital systems were and how hesitant they were to relinquish the information. It was like, oh my God.
Some of these, some of the walls they break through have nothing to do with science. It's just like all the red tape and bullshit that an org that gets to a certain size is just not going to fit through those cracks, and they're never going to even bother, because legal is going to shut down innovation before anything actually moves. So this one-to-end thesis, which I've been pounding on for years,
it's like your life's work.
Finally, and then there's a company that was funded by 8VC, $83 million seed. I can't believe a fucking seed is 83. I was like, did they get an order of magnitude wrong? Like, wait a minute, that used to be a Series B in my day, back in 2016. But like, they raised $83 million to basically, I think, it's just a bunch of BioMarin execs who, not just, like, obviously very accomplished, but now they're getting their piece of the action. And like, they hired a young gun, a CTO, young founder, kind of, tech bio type. But, it looks great. They say nothing specific, but like, this tells you, and their whole language is like about lean. And it's just like, it's kind of gratifying to see all this language I've been using for years, like shouting into a fucking cave. And then all of a sudden now it's starting to get hot. And then if the priority review voucher program gets renewed, which it looks like it got tucked into the latest, appropriations, which is like, this unseemly kind of omnibus bill where lots of shit is thrown in. But this is a piece of pork that I think is good, because if you get the priority review voucher back, and you can basically develop a medicine for a few people and qualify for a voucher that could be worth north of 150 million, close to 200 actually by latest trades, then that's where this becomes not just like, oh, a cool, like, buzzy tech story. It's like, this is the beginning of consumer bio, as some people are calling it. And like, it's starting with these edge cases, like only the rich could afford cell phones back in the day. They used to be the size of, golf carts. And now, like, everyone has it in their pocket. So the same exact thing will happen. I don't think it's going to take 40 years. You're going to get massive compression with AI. I mean, Ryan, I don't know, Jillian, you guys are using AI, whether it's videos, whether it's putting together a patent that we just filed. Like, it is sick, like how just in the last few months,
I was going to say, I was telling somebody that I don't think Pump Science could have been, like, I don't think we could be where we are if we would have like tried to put all the pieces together 2 years ago, because the amount of information and resources and team, and just to even make science easy to understand, and just to make science like entertaining, and just to like build out some of our UI stuff, like, you would need an army of people. And the way that you can use AI now, I mean, to your point, from patents to design to creative to, like, honestly, anything, really and truly anything. I mean, when we were talking earlier about downloading all of the data from Pump Science across all of the compounds, there's probably so much analysis you can use AI for to look across, do a cross-cut of.
We're doing it.
It's crazy.
Carlin will kind of drop it, but like, and at some point, it's just like in a few months, it's getting, even if we're really in the steep part of the exponential to AGI, which I do think AGI, I think we're touching AGI right now. It's kind of like, I like to say it's like low tide, versus high tide. So we're at low tide, and so you got to get really close to the shore to feel it. You got to get right to the edge and push things. But the tide's coming up, and then a fucking tsunami is about to come. And like, it's crazy. It's just crazy.
I think it's so, what I love is like the gift of being on the bleeding edge. It can feel at times like insanity because just when you've gotten your bearings, the tide is making the sand move underneath you like every single day. And just when I think I've like understood resources or how to best use the tools, the sand shifts. And then it's like, oh, I got to go learn XYZ in this tool and in this way. But I feel blessed to be, professionally working in a space where I can use and like figure out how to build with these tools because, previously I was in a Fortune 500 big pharma company and I know they're using AI very much so, but at the same time, like how I'm using it day to day and how we're thinking about solving problems is very different than if I would have had like a corporate desk job. So I feel so fortunate to be like really thinking about big problems and how we can build tools, solutions, platforms, and like products with these resources available. But it is, I will say, like maddening because there are times where it's like, I just got my bearings and I can't even keep up with the amount of intellect that is becoming available to me every single day. I mean, it is truly insane.
I mean, I'm feeding it science data. Ryan is like, you can spare your voice, Ryan. Like it's Carlin's been doing it. It's just sick. Like, and I think, the reality is, you guys are sitting on a gold mine of like longevity data. Like just from our fly results, we're seeing things, and it's like there's no other literature about this. We're going to publish it. Obviously, we're already kind of publishing it on X already, but it's it's just insane how quickly things are can be built in a science first way. Because I can consult with a chemist or a material scientist with PhD intelligence that will talk to me 24/7. Eventually I gotta pay more. I feel like my kid who's like begging me for Robux cards. Like I keep tapping out of my credits and I'm like, fuck, I gotta buy another $200 of credits. Okay, let's go.
It gets you, but I'm hugged. I'm like, oh my, fine, whatever it takes. Yeah, I think what's cool too is that like I think a lot about this in terms of like how people have approached media. Like media used to be very much like centralized and you watched the news, where now it's really gone back to like individual, independent. People are following much more like people for news as opposed to like a CNN or, wherever you watch your news. And I think this consumer bio movement is super interesting because I know there will always be the big players in the space, but with the information and the resources and persistence and intelligence, there's no reason why there can't be this, grassroots movement as well. And what these tools cannot take away from us is knowledge. And so like when you're equipped with knowledge, man, you can move, you can fly. And so before it was like all of this was in little pockets, and when it's at your fingertips, game on. Like it's really at the speed of your idea, which is just crazy to think about. It really is crazy to think about.
Totally.
We'll see. I know it's cool to hear that was a big. Oh, go ahead, Ryan.
And Jillian, I was just going to share, like, I remember this feeling, right? And so it's kind of hard to share this in a way. It's like, it's a feeling, so it's not an easy thing. So Ethan and I knew each other back in the early days before I think the last wave of biotech came back, right? So Ethan had just kind of finished at Harvard. He would just move to the Bay Area. He was doing consulting. I was trying to get this thing called Indie Bio up and running. It was like, no one wanted to fund it. I was like, hey, scientists need money. Oh, no, scientists don't want to build companies. They just want to do science. And I remember, like, I pitched anyone I could that would talk to me in the Bay Area. Peter Thiel nearly funded us, very interestingly, but he wanted to get rid of the lab. And I was like, no, that's the whole point. And then I talked to Sean O'Sullivan, the SOS and SOSV, and he got it immediately. He understood accelerators. I did not understand accelerators. We started Indie Bio. It was crickets when we first opened. No one it seemed like a flop. It was just like, oh my God, there's no one here. And, Ethan and I were talking back, and, it was hard to get access to any kind of resources, but we were making do with the resources we were able to get. Right.
Yeah.
And then, a couple months, whatever, there's always like something that sets off, and then everything runs. And
Yeah.
so it feels like that again. It feels like we don't have enough resources. No one's interested. You know, a handful of weirdos are interested in DeSci. People make fun of me for talking about DeSci. The VCs that I know who are excellent VCs won't fund us, won't fund you. They won't cut us a check to save our lives because they don't really get it. They don't care. They don't get it. And it is one of the most transformational technologies for biotech I have ever seen. And yet here we are, you know, so it's like,
Yeah, it's crazy because I don't know if this ever happens to you guys, but I'm like afraid to go to sleep because I'm like, what am I going to miss? Like, there's so much work to be done. Like there's going to be like some new idea, some new tech, some new thing that could think about how to make this thing go even faster. And it's like, oh, I can't stop thinking about like, Benji is incredible. He's been like, we need to build our faces off because like you just have to keep running as fast as you can because like you said, that moment will be here and you need to have everything built and ready for when you know the big lights come on. And like
you might have been on the money. You might have been on the money. It might have been Musk's comment at the World Economic Forum. That comment that he made that, yeah, I think Musk is like that. He goes, yeah, I think aging is solvable. I just haven't put my mind to it. It's probably a programming thing, you know, like reversing the age or something. That comment that he made, Musk, I've watched Musk long enough, and I've known people who know Musk that when he mentions something, it's not like most of us will say something like one off and go like, oh yeah, you know, here's my comment on it. When Musk mentions it in the past, he's actually said, I have no interest in longevity. That was not the response that he gave this time.
Yeah.
And so that means he's thinking about it. And I think it'll just become this thing as the Optimus robots kick off, as SpaceX does all these other things, as Neuralink keeps going, as you know, all of these other Musk industry companies go, he's going to start a longevity company. And when he does, that will start an entire push into the longevity space. Like I'm just sense, like,
right? Because Sam is behind New Limit and Retro.
Brian is behind New Limit, and then Sam is behind. Yeah.
Yeah. So it does make sense that he's gonna do some. Yeah, it's gonna.
He's
be
coming.
Like
he's coming.
Well, and if you think about it, like all of how he like builds is very much like independent, but it's all actually interconnected. And I can't remember what I read the other day, so I'm not even going to try and quote it. But it was like, obviously, when you go to space, aging slows down, and then when you come back, it speeds back up. So like, there's a huge incentive as you think about like, wait, like if you get truly this movement between, you know, planets. Like, I do think that is a mechanism we must figure out because it's like, well, then you're also creating this like aging inertia where it's like, wait, as you go between planets and you know, within your body, like what is going on? How, you know, it might make sense to go, but does it make sense to come back? And how are the systems working? So to me, it was like
our bodies are going to have to evolve to tolerate a different. You know, we'll be obviously dependent on artificial environments, but there's still going to be the question of radiation exposure, which is longevity limiting. There's still going to be other things that we're going to have to optimize.
You can't go to space unless you cure cancer. Sorry, like literally, this is literally what everyone I talk to is into space. I'm like, I love space. I want to go to Mars. I want to go there. I'm dying to go. However, you can't go unless you cure cancer. Sorry, you can't.
There's going to be a lot of optimization to our carbon blueprint that's going to enable, it's going to be continuous too. And, don't forget, we've got gene editing that's going to go mainstream. We're going to have to make certain edits to make us more resilient. Clearly, like there's a lot of interest in solving the longevity problem, unless Elon just thinks we're going to be mind melded with the robots and we're just going to move on to silicon from carbon. But hopefully he's not too ideological about that.
To me, whenever I have questions about what does Musk think, I just go back to the Culture series, the series of books from Ian M. Banks. Most of his insights, some of them come from Asimov. Most of them come from the Culture series.
Good point. Yeah, that's a good point.
He's gonna go after longevity. I'm certain of it. Like that comment he made at the World Economic Forum, I saw it too, Jillian. The moment he kind of stopped and said, "No, I haven't spent much time on it," that's very Muskian. Like, he is going to come back to it, and this category will explode. And we are positioned in this space where it's about to explode. And it's going to explode because of AI. Like, what Ethan, what you were saying, like these N of 1 companies, biotech companies, Sam Altman's showing it. He wants the IP. He knows where the value drivers are. We just filed IP we developed with multiple AI systems and pump science data, right?
Yeah.
So like when we submitted our provisional for rapamycin SMR28, which Ethan was working on 20 years ago, the data that we used was in worms, flies, and mice, and we were able to put that in as a provisional. That is an incredibly strong provisional patent.
What a package.
Right?
What a package.
Mm-hmm.
Yeah,
Mm-hmm.
That's crazy. That is crazy.
This is the beginning. Like, I can't wait to see all these other companies filing with data that was built on the pump science platform.
Yeah. These conversations give me like I said, I'm having a hard time sleeping, but these conversations, like when you think about
Jillian, we all are either babies or just insanity in our heads.
Insanity. Right? I've always said I'm like a little off center, like it's like, just like a little. My husband's like, what are you doing? Like, you need rest. And I'm like, there's so much work to be done.
Hypomanic. I tell people I'm hypomanic, which I actually think I am. I'm like just below manic, like the energy levels are high enough. Ethan, I don't know if you've ever explored this, but you have a lot of high energy too. So we all have something that's not quite right that drives us hard.
It's like a little off center, but it's deep. I mean, you like can't shut it off. Like, it cannot. I have one speed all in, and it's like intense. And there's just so much work to be done for, like, I mean, I have kids, and I just sit and I like look at. They talk. It's kind of fascinating how kids like talk about death, and they're like, "Are we all going to die? Are you going to become a grandma? When are you going to become a grandma? Every time I have a birthday, you're going to get older." And like, to be able to honestly like know what I should be taking, take it, see results, and then leave that information for them someday. Like, there's no greater, like, to me, mission to be done here, because like this information is not readily available. I don't want to have to take 75 supplements in the morning and not know which one's actually working, and then leave that information for the next generation. But I'm just so grateful to be amongst such intellect. You guys are incredible. What you're doing is incredible. And I know that we are just getting started. And it's our mission to also get more people in on this mission, because we'll only be able to keep building if we can get more people to see and believe in what we're doing.
Jillian, I just keep thinking what Musk said. He was like, "Hi, I think it's actually like very doable. Today, we know we can extend the maximum lifespan of multiple species by 40+%. Why don't we have those therapies in humans? I don't understand. Like, I don't understand. We know it works in multiple species. It'll work in humans. Maybe we don't get 40%. Maybe we get 10% or 20%.
I'd take it.
But
I would
there's
take
no
10%.
additional science necessary. It's literally the stuff that we have the data on. And so it's like, Musk is right. It's just a matter of time before the machinery that's focused on AI and robotics and space finally moves over to longevity. People talk about longevity. Yeah, Peter Diamandis is into it. Some people we haven't yet seen what's going to happen when all of Silicon Valley, all of Wall Street say, "Oh, wait a minute, longevity. This is big."
Yeah. I mean, it really and truly, like when you think about the barriers that exist, and I don't even know that barrier is the right word. It's just these processes have been set up on archaic systems, and this industry is just ripe for innovation. And, I think you kind of have to play outside of the lines a bit to like show people how the system could be, like this founder mode on your cure. Like, no one can design that system until they're put in that situation, and you have to it's survival mode for them. And then you take those learnings and you kind of string together alternative systems. Same thing for us, coloring, really like thinking outside of the box, because if you color in those lines, it all leads to roadblocks and kind of the same. It's just like an impossible hill to climb. It's just an impossible hill to climb, which is why only so few people get to do it. And so we must reimagine these systems. We must find newer and faster ways with the resources we have available, because humanity, our lives literally depend on it. Like I'm still dying today, so I have a lot of work.
As are we all. I mean, my
I got
throat.
a lot of work to do.
Yeah, it's like. Yeah.
I mean, we should do this more often. Should we do like Friday chats? I mean,
I think so.
this
This is a
good.
cool. This is a cool thing for us to do because it's something that it brings in. It's not just about one company or one drug or one initiative. It's like a much broader perspective on where we're going as a biotech industry, as longevity, like the thing that matters to us most, right? Which is longevity. Longevity is everything. We treat diseases of aging when we should be treating longevity. It's like, it's a really interesting problem in our medical system. Like, you know, your cartilage fails. Oh, now you're getting osteoarthritis. Oh, what if we had functional partial reprogramming? You wouldn't have that problem. And what else wouldn't you have if you had that?
Yeah.
Right?
And like, I think the fact that we have become okay with like incremental benefit is benefit enough. And like, once you have a problem, like I find it so fascinating that medicine is really a checklist. And until I think you're in like a health situation, do you realize like doctors follow a checklist, and at the end of the checklist, it's like, I followed the protocol. There's nothing left for you. And so what I think is fascinating is it's like, yeah, but like, how do you even think about things before you get into the protocol? Or like, why isn't there a protocol before the protocol? Like, you know, a lot of it is just like disease and symptom management. And like, I want to know what like the health protocol is. Like, how do I build the thing so I don't get in the thing? And yeah, it's just, I've been very humbled. I think a lot of like COVID really showed me how protocol-based medicine is, as it should be, you know, as it should be to get desired outcomes. But ultimately, when you're thinking about a different protocol, or you're at the end of a protocol and there's nothing for you, that's when you really realize like, wow, we got a lot of work to do. I mean, like our mission is just getting started for sure. Guys.
All right,
Well, I say we run it back.
We
run it back. Yeah, these are good. I was also, you know, I'm trying to
we
be
need
better
to
about.
move away from the local maxima of treating the diseases of aging and treating aging itself to extend health span and lifespan. Like it's just becoming clearer and clearer to me every day. And honestly, as I get older as well, like we're treating like, oh, that bit fell off. Oh, that bit is not functioning right. Like what are we doing here?
What are we doing here? Why are we letting this happen? Yeah. Why are we letting, you know, I think it might've been Ethan who said it, where it's like, you have all these millennials that are like, that are coming upon this moment, and they're like, what? Like, this is what's like, I'm supposed to just gradually decline from here. And with their, you know, tech experience and, you know, maybe like economic situation, they're able to just like refuse to accept that is how it will be. And, you know, I would love to bring that to the masses. Like, I think it shouldn't just be, you know, sit with people who can afford it. Like, I would love to be like, Mom, you know, like, let's make you anti-fragile. It's not if you're going to fall, it's when you're going to fall. This will help you help you stay anti-fragile.
Raptor treated flies don't fall. They keep climbing.
Keep climbing. I mean, like this is how we should be thinking for sure. Well, guys, I say we run it back. Maybe do these more often. I was also streaming on LinkedIn. I think there's a huge, you know, wealth of curiosity over there as well. I think part of what we have.
I agree.
Have to do
LinkedIn. I actually shared the LinkedIn. I shared this stream to LinkedIn, and it's a long tail. So LinkedIn is a long tail, whereas X is a short tail. You know, X. Yeah. If it's hot, it's hot. If it's not, it's dead. LinkedIn it's hot over time. Yeah.
I think these conversations are very much like happening over there too, in a different way, and being better about, you know, bringing the science, the approach, the ideas.
More, it's more the people we want to reach, right, Jillian? I mean, it's really the people who are doing the research, who are in the labs, the scientists, the entrepreneurs. The biotech people are more on LinkedIn than they are on X. Yeah. They should get on X. So if you're listening on LinkedIn, you should get on X. It's important. I know you probably won't, but you should. You know, the tech people, I know that they dominate, and the crypto people, you should still check it out.
Yeah. I'm a big believer too in training your algorithm. Like if you're really intentional about what you like, if you go on there and you don't get distracted and you engage with the right things, it will serve you what you're looking for. Just I am guilty of being like, oh, I found myself, and it was like, no, now I'm getting this type of content. And it was a one-time thing. I didn't want that all the time. But you can train the algorithm too. But yeah, I would love to run these conversations back. There's a whole just even coming out of this like data drop, thinking about the studies that are coming, what you guys are doing. I have like 75 topics. I could have just been like, hey guys, also that we could have continued to talk about. So let's do more of these. I'll write them
Sounds
down.
great.
Cool.
All right.
Sounds good.
Awesome. Thanks
Thank
for
you.
stopping by, guys.
Yeah.
Have a great
cheers.
weekend. Bye.
Bye.